I think I can speak for all of us here today when I say we're all intimately familiar with lipid nanoparticles, right? No? No? Well, you should. You've likely already had these particles inside of your body in the form of the COVID-19 vaccine, which was the single largest and most successful health campaign in human history, showing the power of these new mRNA therapies to revolutionize the way we approach modern vaccine science. Now, the way these vaccines work is actually fairly simple. They take a very small fat bubble, around a thousandth the width of a human hair, and they load it with mRNA that contains instructions that tells the cell to make a single specific protein. For the COVID-19 vaccines, this encodes for the spike protein of the virus, which then informs the immune system to recognize the virus and prevent infection in the future. Now, despite the unprecedented success of these vaccines, they have one major flaw, and that is that they must be stored at ultra-cool temperatures, on the scale of -20 to -80 degrees Celsius. For reference, this is less than negative 100 degrees Fahrenheit. I'm from the Midwest, so I can tell you that's very cold. At these temperatures, they have shelf lives around 6 to 18 months. However, they must be kept at those temperatures from the time it is made in the factory to the time it is given to a patient. This is known as the vaccine cold chain, and it is incredibly expensive not only to purchase the freezing technology required at each step, but also operate it for years on end. Many low-income countries simply cannot afford to maintain the cold chain, and so it breaks with devastating consequences. If these vaccines are allowed to reach room temperature, their shelf lives plummet to less than 24 hours. What does this amount to? Between the peak of the pandemic and 2023, 5.6 billion doses of the COVID-19 vaccine went to waste. This represents nearly 30% of all of the vaccines made in that same time period. And more than $100 billion of government money down the drain wasted on vaccines that never made it to patients. So how do we solve this? Well, our lab came up with 3 new approaches to vaccine design to overcome these problems. 1, we want to freeze-dry vaccines. This process removes the liquid the vaccines are kept in, providing a much safer environment for long-term storage at all temperatures. 2, we want to load the mRNA onto the surface of the particles rather than putting it inside. This will not only enable us to freeze-dry these vaccines without losing efficacy, but also enable us to add any mRNA we want to these particles instantly through simply hand mixing. And 3, we want to co-deliver this mRNA with other therapeutic drugs. This will enable a whole new spectrum of vaccine design that was not before possible. So to accomplish this, our lab designed these hybrid lipopolymeric nanoparticles, which combines the material benefits of both lipids and polymers. You might be asking yourself, what is a polymer? Well, broadly, a polymer is any long-chain molecule with repeating units. And that might sound complicated. However, you interact with polymers in your daily life, from the plastics in your water bottle, to the materials in your clothing, to the wood in the chairs you're sitting in, or to the proteins and DNA in your body. And here we utilize these long-chain synthetic polymers because these chains can entangle with each other and entangle with the lipids in the creation of these nanoparticles. We can then freeze-dry these particles into a shelf-stable powder, which is something you cannot do with lipid nanoparticles without significantly harming them. Next, we can reconstitute these powders in water. And in solution, the particles have an overall positive charge. mRNA itself is inherently negatively charged, so just by simply mixing it by hand, they will instantly bind with each other, and we can have a vaccine ready to inject in less than 5 minutes. Furthermore, we can load therapeutic drugs into the initial nanoparticle mixture, allowing us to co-deliver these drugs with the mRNA all on one particle. So what do these particles look like in the real world? You can see here a picture of the particles both when they're freshly made, after we freeze-dry them, and after we reconstitute that powder in water. Zooming in on these particles using an electron microscope, you can see that they're spherical and roughly 100 nanometers in diameter, which is very similar to the particles used in the COVID-19 vaccines. To prove that these particles can be used to deliver mRNA to cells, we utilized mRNA that encodes for green fluorescent protein so we can visually observe when these cells begin making this protein. You can see here that after we add our nanoparticles to these cells, we see a high amount of widespread degree of expression, showcasing that our particles can indeed not only deliver mRNA to cells, that this delivery results in functional protein expression. Quantifying it, you can see that our particles perform at the same level as the lipid nanoparticle technology on the market today. Now, making cells glow green is interesting and all. However, we wanted to find a new avenue of therapy that could be unlocked by our nanoparticle technology. So we designed this dual therapy approach in which we deliver both an mRNA and a chemotherapy directly inside of a tumor. In the first stage, the particle enters the tumor cells, releases the mRNA, and the tumor begins producing these inflammatory proteins. Essentially, we're hijacking the tumor to make these proteins for us. In the second stage, the particles release the chemotherapy and kill some of the cancer cells in the tumor. However, many times, when you apply chemotherapy to tumors, not all of the tumor will be eliminated, resulting in a recurrence down the line. This is where the dual approach comes in. Those inflammatory proteins from before will activate immune cells surrounding the tumor, which then go on the attack and clear what's left behind. To examine this effect in the real world, we injected tumors onto the back of mice and then treated them with our particles. You can see here that the untreated tumors grow rapidly out of control. However, when we apply our therapy, the tumors grow 3 times slower, making them much more manageable for treatment. Not only this, after 19 days, all of the mice in the untreated group had died. And after 21 days, the majority of the mice in the group that just received nanoparticles with chemotherapy also died. It is only when we applied the nanoparticles that had both the chemotherapy and the mRNA that we see 100% survival in that same time period, showcasing a real-world example where we can see actual benefit in diseases with these nanoparticles. So I hope today I've been able to demonstrate to you why my research matters, and how the nanoparticles that we're developing in the lab today can not only overcome the current issues we're experiencing with lipid nanoparticle technology, but build new strengths in the way that we develop the next generation of vaccines. Thank you.